Why recursion uses images of cells is quite interesting, actually. There's a lot of biology happening inside any one cell. And if you look at any one target, one gene, one protein, that's usually not telling you the whole story about what's going on. Even if you look at something like overall growth or death of cells, there are lots of different ways that cells can grow, lots of different ways that cells can die, and they don't always look the same. So for example, you run into situations, particularly in anti-cancer drugs, where you make a new molecule as you're optimizing a series, the cells die, and you think, we've done really well, but it may be that your new molecules are actually doing something completely different in the cell than what you intended. So getting a broader picture of what's happening inside of a cell has been one of the big trends, really, of experimental biology in the last 15, 20 years, whether you're talking about technologies like sequencing of DNA and RNA, whether you're talking about technologies like proteomics, looking at many proteins inside of a cell rather than just one protein at a time, or what we've chosen to do, imaging. So it turns out that cells, when things happen to them, will actually literally look different. Sometimes you have cells that are round and look really happy, and under stress, they might become more and more elongated. Or they might, start to stop having smooth edges and start looking really frilly and weird around the outside. Or there might be even more subtle things that are going on, things that are subtle enough that you wouldn't be able to see them by eye. Now, another one of the challenges is beyond the things that are subtle. When you're taking pictures of things, you can have lots of weird stuff happen that we call batch effects, right? That are irrelevant to what you're looking at. Thinking about taking a picture of the same person as a cloud passes in front of the sun. It's the same person. They're not probably any happier or less happy second to second, but the lighting in the scene completely changed, and that's going to look like the major difference in the image. Now imagine that you're playing out in the park and you take a picture of somebody and then a cloud passes in front of the sun, and at the same time they got hit by a water balloon. Well, you might actually see that the main change that happened in the image is that it got a lot darker, but the thing that you actually care about is they're very angry because they got hit by a water balloon. And you would miss that because of the gross change. And so the big challenges in dealing with this imaging data, although there's a lot of information in it, although you can tell, does this person look happy? Do they look tired? Have they gone for a run? Are they relaxed? What's the background look like? It's not structured in a way that it just immediately tells you, oh, here's the image and here are all the things that are going on, and I've gotten rid of all the irrelevant background information to tell you what you need. That's meant that other technologies like sequencing have become very, very popular in the field. Because if you sequence all of the RNA molecules inside of a cell, you can actually take that and you can do a process called alignment, and you can find out, oh, okay, well, gene 1 went up between these two conditions, and gene 2 went down, and gene number n, that stayed about the same. It's a nice tabular form that's easy to understand, easy to analyze. It's called structured data. But when you're taking images of cells, these are just pixels. They're not structured. And so it ends up being a difficult thing to actually do analysis on. Think back to, if you had, an old iPhone 3G or something, where you had a camera, but you wanted to find a picture of your dog. You're just having to scroll through the camera roll forever until you find that specific picture that you want. Today, you can go into the Cameras app and you just type in the name of your dog or the name of one of your friends, and it'll immediately pick that up, because we have algorithms now that have been able to structure the unstructured pixels of these into meaningful elements that are useful for us to use. So what recursion wants to do is actually structure the unstructured, rich information coming out of these images in order to perform biological discovery. Now you might ask, okay, well, why do that? We can use another way of getting information that already comes out to be structured. Well, the short answer is there's just as much, if not more, probably on par, the amount of information that you can get out using a technology like this. And this costs you 1/10, maybe 1/100th as much to run the experiment. And what that means is that for the same amount of money, you can collect even more data. You can assess a broader swath of biology. And that's very interesting. It just means you have to solve this one last problem of how you actually structure it. The other thing that it lets you do is that there are some things that you just can't measure in this way. So for example, if you're going to pull all of the RNA out of a cell in order to measure it, well, there's no RNA, there's no more cell, like the experiment is done. If you image a cell, in principle, you can take a picture and then you can come back a day later or 2 days later and you can take a picture of those same cells and you can actually track how they evolve in time. So imaging affords some very interesting capabilities where we get access to a very broad range of biology. We can do so very cheaply. In principle, you can do so in a time course manner, but you just have to solve this one problem, which is admittedly a big one, around how you structure all this unstructured, noisy information. So a big piece of where Recursion has invested in machine learning over the more than 10 years that we've been around has been in solving that problem. We work on ML problems in a lot of different domains, but this is the one that arguably we've invested the most time in. Now, because this data modality is not as frequently used in the field, in many ways, we've had to lead the field here. We've shared some of the biggest data sets to help catalyze research in the space, the RxRx1, 2, 19A and B, 3 data sets. We've written papers in the field. We work with leading academics and conferences in the field to help accelerate it. But let's talk a little bit about how it is that we actually address this. So one of the keys here is that it would be great to have a model that you can use for multiple things. And this has been a trend in machine learning for about the last 5 years, to build what are called foundation models. The notion of a foundation model is that it is something that can serve as the foundation for more work, more things that you would build on top of it. There are a lot of different tasks that we might want to solve in machine learning. And for a real world example, think about going to something like ChatGPT. You know, when you ask it a particular question, it has probably never been trained on exactly the question that you asked it. What it has had to do is learn from a broad swath of natural language and other information how to generally complete the range of tasks and perform well on them. And foundation models are similar to this. The question is, how can you take a large amount of data in, generally learn the structure of this into something that we call a foundation model, and then use that as a lever for new tasks that you might want to run downstream? So that let's say that we're trying to do a new kind of prediction task, but we only have a little amount of data on it. Well, in a traditional machine learning context, this little amount of data may not be enough to actually do a good job of that prediction. The goal is to have a foundation model that can act as a lever, in the sense that a very small amount of data can actually perform as well combined with the foundation model as a much larger set. And this is something that we've been seeing in our own work, in building foundation models, both for imaging as well as in areas like chemistry. Now, this is important because the space of possible biology that you can observe, the space of possible chemistry is incredibly huge, but getting that data still tends to be slow and expensive. So you want to build as much as you can to break these scale barriers. So how do we do this in the space of imaging? Well, a general strategy for building foundation models is a machine learning strategy called self-supervision, where you may not have a ton of data where you've exactly labeled every instance of every class. That can be very expensive. If you ask someone to go through the entire camera roll in their phone and hand label every person in every picture, that would take forever. What you can do is you can ask them to label a few of them, but really you need a way to learn from unlabeled, unstructured information. So a general task that's proven useful in the self-supervised context is predicting things that are missing. If you look at things like language models like ChatGPT, the task is, given a sequence of words, word 1, word 2, word 3, what's the missing word? And that's something that you can do at scale from data that nobody had to look at, because you can take a whole article and you can just chop off the end and say, okay, well, given the text that came up until this point, what's the next word that ought to complete it? We do something that's very similar with the images that we build here. So imagine that we have an image, and what we have in here is a bunch of different cells. What we can do is tell the model, we're going to give you an image, we're going to give you an image where we've randomly blanked out particular parts of the image, and we'd like you to guess what fills in those gaps. And it turns out, remarkably, that models can do this very well. Now, this kind of makes sense, right? If you had, for example, a picture of a face. I'm terrible at drawing faces, so I'm terrible at drawing generally. You know, you can imagine that if I told you, okay, well, I'm just gonna mask off this bit here in the middle, you could probably guess there's going to be a nose here, right? Now, you might have trouble telling exactly how big that nose is, exactly how it's shaped, but maybe depending on the rest of the characteristics of the face, you'd be able to fill it in reasonably accurately. It turns out that these models, called masked autoencoders, can do a very similar task on these images of cells. Now, what I've drawn here may look, fairly unsurprising. That, yeah, okay, sure, you blank out, what looks like maybe 5 or 10% of the image, it can guess the rest. The really remarkable thing is that you can actually blank out something like 75 to 90% of the image, and they'll actually reconstruct it very well. And in doing so, what they've done is they've learned something about the intrinsic structure of images of cells. The same way here, that we took advantage of the knowledge that we have of the structure of images of faces, that what ought to be here is a nose. It probably shouldn't be a finger or a beak or a trash can. We knew that there's structure to the images of faces. The models learn what the structure of images of cells are, and they can learn some parameters that tell it about the biology that's going on in the image generally. So what we do is we train the models to solve this masked autoencoding reconstruction task, and then ask the model, given a new image where nothing is masked, where you can see all of the cells, what are the features that you have learned about that image? So we can take this and we can essentially turn an unmasked image now into a list of numbers. Now, what do each of those numbers mean? Don't know. It's not built to be an interpretable model. However, what we can do is we can ask, well, given one of these sets of numbers, and given a database of all the other images of cells that we've taken in the past, how similar or how different is this image, or the conditions under which this image was taken, to the other conditions that we've looked at? And an operation like this, called similarity comparison, forms a root of a lot of the work that we do with our maps of biology, where, for example, we can tell that if we have one gene, let's say that we had one gene over here that's known to be associated with a particular kind of cancer, or a good target for a particular kind of cancer. If we then identify, oh, hey, look, there's another gene down here that looks extremely similar to that, or the cells that have lost that gene look extremely similar to cells that have lost the known target, that this gene might also be an interesting target to target for that kind of cancer. And in fact, that's the story of the genesis of our RBM39 program, where we knew that CDK12 was an interesting target in certain kinds of cancer, and we discovered both RBM39 as well as a compound in our library targeting RBM39 as an interesting way to get into that pathway while avoiding some of the liabilities associated with trying to drug CDK12 directly.